This recording is going to be the first part of a two-part webinar series that covers muscle tissue. We've talked about skeletal muscle already in lecture, so what we're going to be left with is looking at cardiac muscle and smooth muscle. So the first part's going to cover cardiac muscle. But as a quick reminder, is when we started covering skeletal muscle, we looked at different functions of muscles, and what I've done is I've listed them again, but what I've done in red are ones that also apply to either cardiac or smooth muscle. Now, all muscle is designed to contract, so it's going to be the interaction between actin and myosin, and they all somehow are involved in movement. So like with skeletal muscle, we learned about locomotion, manipulation, facial expression, things like that. But cardiac muscle will be involved in pumping of blood. Smooth muscle is actually part of vision in that it can change the shape of the lens and it can change the diameter of your pupils, but it's also involved in processes such as urination and digestion and reproduction. Now the support of soft tissues. Now muscles can help to support the weight of visceral organs and shield organs from injury, and that can apply to both skeletal and smooth muscle. And another thing that muscle does is it guards entrances and exits. So there are some smooth muscles that are sphincters, such as the internal anal and the internal urethral sphincter muscles, which are made up of smooth muscles. So they're involved in processes of defecation and urination, but also precapillary sphincters, which are part of blood vessels, is right before the capillary beds, which can control blood flow through capillaries. So that's also smooth muscle. So what I want to do first, though, is we're going to start talking about cardiac muscle. So cardiac muscle, its location is only in the heart. That's the only place it's located. So if someone says cardiac, you always think of having to do with the heart. Now, its function is when it contracts, it creates tension, which creates pressure, which is used to pump blood. Now, it can pump blood up to the lungs, which will be part of the pulmonary circulation. And what its job is, when it's pumping that blood up to the lungs, that's where the oxygenation of the blood will take place and get rid of carbon dioxide. Now, when it also pumps blood to the rest of the body, which is part of called the systemic circulation. So it's generating that pressure. They'll be able to move that blood through your systemic circulation to deliver things like oxygen and nutrients and hormones to various tissues. So let's look first at the anatomy of the cardiac muscle. Now the cells are typically branched. So you see kind of how here it has these little branches to it. So the cells are branched, and they're usually uninucleate, which means usually you have one centrally located nucleus. So you can add that to it. It's centrally located, because that's going to be different from skeletal muscle, because skeletal muscle is multinucleate, and it's also, the nuclei are found on the periphery, not in the center. Now, like skeletal muscle, the cardiac muscle does have these striations. So you see all these little striations here. Later, they're written here. So it's striated because the actin and myosin filaments are regularly arranged into sarcomeres, just like skeletal muscle. Now, one thing about the cardiac muscle, though, is the T-tubules are relatively short, and there's no such thing as a triad. So the triad in skeletal muscle was a T-tubule and two terminal cisterna of the sarcoplasmic reticulum. But if you look here, these really aren't terminal cisterna. They don't kind of expand out. So they actually refer to them instead of triads. There's no triads. They consider them dyads. So the T-tubules are kind of wide and a little bit more broad than in the skeletal muscle. In the sarcoplasmic reticulum is not as well developed. It's poorly developed in cardiac muscle. And because of this, calcium, which is needed for contraction, is going to be needed from both the sarcoplasmic reticulum, but a lot of extracellular fluid calcium is going to be important with cardiac muscle. Now, the cardiac muscle does have quite a bit of mitochondria, very large mitochondria. A lot of the sarcoplasm is made up of mitochondria when you compare it to skeletal muscle. They also have a lot of myoglobin. So because of this, having a lot of mitochondria and having a lot of myoglobin, which is a binding pigment for oxygen, cardiac muscle does not easily fatigue. Just think about it, your heart's beating all the time. You don't want it to fatigue. So the cardiac muscle relies heavily on aerobic respiration for energy. Now, the cardiac muscle cells, though, also, or we say cardiac myocytes, they have these specialized structures referred to as intercalated discs. Now, and that's classic with the cardiac muscle. If you see an intercalated disc, you know it's cardiac muscle. Now, the intercalated disc actually composed up of two things. The first one is the gap junctions. Gap junctions allow that direct electrical connection between cells. So allows the passage of ions from cell to cell very quickly. And that's going to be very important in allowing cardiac muscle to act as we refer to as syncytium. And a syncytium, a functional syncytium, is where you have the cells are kind of mechanically and electrically connected to each other. Now, the mechanical connection is, again, part of the intercalated disc, and it's the desmosome. So you learned about desmosomes before. So the desmosomes help to kind of maintain the structure to kind of keep the cells from being pulled apart. So the cardiac muscle is like skeletal muscle, that it's striated. It is similar to skeletal muscle in that, in some other instances, but not structurally. So they're branch cells. Skeletal muscle's not. It only has one nucleus. Well, the skeletal muscle is multinucleate. Now, also one of the things that when we look at functional characteristics is cardiac muscle exhibits what we refer to as automaticity. Or I can use the term is myogenic, in which the electrical impulse or the origin of the electrical impulse needed for muscle contraction occurs within the muscle itself. The somatic nervous system is not needed to generate action potentials for cardiac muscle to contract. The cardiac muscle has these cells in the SA node, they're called P cells, or pacemaker cells, that exhibit automaticity. So the electrical impulse begins in the SA node, and eventually it's transferred through these specialized cardiac conduction system cells and to the contractile cells of the heart. And you'll learn more about this in AMP2. But cardiac muscle, although it exhibits automaticity, so it can contract on its own, and it does in a regular, repetitive, rhythmic manner, but the autonomic nervous system can affect the rate as well as the contractility of the heart. So you have innervation by the sympathetic nerves, which you see here. And you also have parasympathetic innervation, which is really via the vagus. So make a note that parasympathetic nervous system innervation is through the vagus, because you should remember that. Now, what's the effect of the autonomic nervous system regulation of the heart? So this is a review, because we did autonomic nervous system. Now, norepinephrine is released by sympathetic nerve endings, and it binds to beta-1 adrenergic receptors located on the SA nodal cells, which affect heart rate. But it also binds to these receptors located on the cardiac muscle, which will affect contractility. Now, the vagus releases acetylcholine. So actually, acetylcholine should be a capital C here. So acetylcholine is the neurotransmitter used, and it binds to muscarinic receptors in the heart. And what it primarily does is it affects heart rate. Very relatively minor effect on contractility, and again, this is something that you will learn about in AMP2. The cardiac muscle, though, is also the contractility and heart rate can be influenced by other factors, certain hormones. We could actually include norepinephrine and epinephrine released by the adrenal medulla. They will also bind to those beta 1 adrenergic receptors, but because these are being released into the bloodstream from the adrenal medulla, they're hormones, so they can affect heart rate. They can increase heart rate and contractility. Thyroid hormones can increase heart rate and contractility, and primarily the means by which they do that is they increase the number of the beta 1 adrenergic receptors on the heart, so it makes the heart more sensitive to the sympathetic nervous system stimulation. Other things that could affect is stretch. So if there is more blood going into the upper chambers of the heart, called the atrium, that actually increases heart rate. And there's just something that you'll learn about called atrial stretch, and something referred to as the Bainbridge reflex. Also, if you have more blood in the ventricles, that would stretch out the muscle, and that will actually increase contractility. And that is something that you'll learn about that's called referred to as the Frank-Starling mechanism or law of the heart. That's something, again, that you'll learn a lot about AMP2. Now, temperature also affects heart rate. So higher temperatures increase heart rate, lower temperatures decrease heart rate. So the heart can be affected by a lot of different factors, although the initiation of the electrical impulse is within the heart itself. Now, the cardiac muscle is different than skeletal muscle in that its time in which it contracts is a lot longer than in skeletal muscle. When I just think about the heart needs to be able to generate enough pressure to be able to pump blood. So I want to spend a little bit more time contracting, but it also when you look at the refractory periods, so this light blue refers to as the absolute refractory period. The yellow represents the relative refractory period. The refractory periods in cardiac muscle are much longer than in skeletal muscle. So what the end result of that is you can't produce tetany, or you cannot produce tentanic contractions in cardiac muscle, which is important because if it's in tetany, it's not going to be able to relax and fill with blood because it needs to fill with blood to be able to pump it out. Now, another thing that's different is, it's a slight difference, is the excitation-contraction coupling in cardiac muscle. So this is a good time to kind of review a little bit of excitation-contraction coupling in skeletal muscle. So in skeletal muscle, we talked about the generation of the action potential and how it was propagated down these T-tubules. And then what it does is it activates what you see number 2. This is the DHP receptor. Now in skeletal muscle, that DHP receptor was a voltage sensor, which normally blocks the ryanodine receptors on the sarcoplasmic reticulum. But in the case of cardiac muscle, is that when the action potential goes down the T-tubule system, the DHP receptors are actually calcium channels. And what you'll see is you notice this calcium coming from the extracellular fluid is actually going to go into the sarcoplasm. So remember that the sarcoplasmic reticulum is not as well developed in cardiac muscle, so it's going to rely on more calcium coming from the extracellular fluid. So you'll see that this calcium is going in through the DHP receptor, which actually then activates the ryanodine receptor. The ryanodine receptor in cardiac muscle is calcium gated. And so that calcium is going to activate these ryanodine receptors in the sarcoplasmic reticulum and release calcium into the sarcoplasmic reticulum. And everything from there on, though, is the same as we talked about in skeletal muscle. Calcium will bind to troponin C that causes a conformational change, which then moves tropomyosin out of the way of the myosin binding site on actin. The only difference is what you're seeing right here. Okay, so let's do a little quick, quick comparison between skeletal and cardiac muscle. So the trigger for the sarcoplasm or sarcoplasmic reticulum to release calcium in skeletal muscle is voltage. In cardiac muscle, it's calcium. The DHP receptor in skeletal muscle is actually a voltage sensor, which is blocking the ryanodine receptor. But in cardiac muscle, it's actually a calcium channel, which allows extracellular, or I should say calcium from the extracellular fluid to go into the cell. The ryanodine receptor in both muscles are calcium release channels, but in cardiac muscle, it's calcium gated. In skeletal muscle, it was blocked by DHP, the DHP receptor. Now, the calcium released by the ryanodine receptor, the amount that's actually released in skeletal muscle is directly related to voltage. It's proportional to membrane voltage. The greater the voltage, the more calcium that's going to be released. In cardiac muscle, it's proportional to the amount of calcium that's entered. Okay, so there's a little bit of a difference there. So that's the main things that I want to cover for cardiac muscle. The next recording is going to look at smooth muscle.